In the next hour or so, I'll start a little bit of pluripotency. You've worked with the cells already. I want to talk about their unique biology, just touching on some of the history of what makes them pluripotent and unique, and then they want to leave pluripotency, and you're about to not let them leave chaotically, but leave in a directed differentiation manner. Directed differentiation is the term we use to summarize the controlled differentiation you're about to induce with adding growth factors to the media. And directed differentiation means the recapitulation in this in vitro model system of known in vivo developmental milestones. So we're going to talk a bit about development today, or the first moments of development. So let's start right in. This is going to be a chalk talk, so forces me to go nice and slow so everybody can keep up, and I get to smell magic markers along the way, which is really most of the attraction for me. Okay, so the moment you're starting with is the fertilized egg that can be done in a fertility clinic or in the fallopian tube, right? And at that moment, you're a unicellular organism, and that is how we all began life, right? So if we start at the unicellular stage, we can call that cell totipotent, obviously, right? It can become anything, extraembryonic, intraembryonic, placental, all the cells you are coming from this one cell. This is a totipotent cell, ex vivo or in vivo. This is not what your embryonic stem cells are. So if you look at the first few cell divisions of life before the human embryo implants in the womb, so traveling down the fallopian tube, we've got a cell division to make 2 cells, another one to 4 cells. Before you know it, you're a ball of cells. That's the morula, right? We think at that place, the morula, you can separate different cells and still form the entire organism and extraembryonic tissue. So we're still totipotent through this stage. Okay, and if we arrive in the mouse at something like 3.5 days post-fertilization, we now start to have some asymmetry, all right, and you have these embryo formed, a ball of 100 or so cells, and there's a fluid-filled center, and at this point we have the blastocyst embryo. Whether you're a human or a mouse, they don't look identical, but they're fairly similar, and the biology should be similar. And now you don't have totipotency anymore. You have some segregation of potential and gene expression, and these cells around the fluid-filled cavity form trophectoderm and your placental tissues, and the embryo proper, or all the cells you are right now in your body, are coming from this region here called the inner cell mass, ICM, inner cell mass, and those cells we call pluripotent. What does pluripotent mean? It's a scientific definition for our purposes in this course and in this embryo, is the ability to form more than one cell type from more than one germ layer. So if you're forming all 3 germ layers, all the cell types you are in the body, in the embryo proper, we call that pluripotent. And if you've lost the ability to form everything, if you've lost the ability to form placental tissues, then we're no longer totipotent. So somewhere along the way, the biology is switching, and this inner cell mass state of pluripotency is not a state of pluripotent stem cells. So just to be clear, in you right now, after you're born, we don't have any evidence that you have leftovers of inner cell mass-like cells that have the same biology or the same pluripotency, and therefore they're not self-renewing, and we don't refer to them as pluripotent stem cells. They're just pluripotent, and within hours of this moment of life, they make cell-fate decisions, or they differentiate, so they lose this basal pluripotent state, so they're not self-renewing. So stem cells, you may have touched on yesterday, are defined by the ability to indefinitely self-renew as well as differentiate. Certainly these cells can differentiate, but they don't indefinitely self-renew, so we don't call them stem cells. They're pluripotent cells. Before we get later in development, which is what you'll start to induce later today, let's just talk about the pluripotent state and what makes this unique and interesting. So if these cells can form all the cells of the organism, in vivo at least, it's about to implant in the womb in a moment, this embryo. The interest in the field, obviously for decades, was can you capture this normally transient state in nature in a laboratory dish to do interesting biomedical research. And so that was a long struggle that culminated in 1981 in the seminal discovery of the in vitro techniques that allowed capture of these cells in a stable state in culture. So there were 2 back-to-back, or not back-to-back, they're different journals, but 2 near-simultaneous papers. One was in PNAS by Gail Martin, a single author paper, and another was by Evans and Kaufman in Nature. And both groups figured out after a long struggle and race that if you could culture mouse blastocyst embryos in precise conditions that happen to be, in one case, conditioned media from teratocarcinoma cell lines, early pluripotent cell lines, that they could capture and grow out from the inner cell mass cells of a stable karyotype that could be passaged, that could retain pluripotency after passaging. And now that gave birth to the term pluripotent stem cells, because the cells were no longer just pluripotent meaning they could be put back into mouse chimeras or mouse embryos or differentiated in vitro, but they also could be passaged in a stable state provided they were either on special feeders, which we'll talk about in a second, or in these conditioned teratocarcinoma conditioned medias, and therefore they were undergoing self-renewing divisions to maintain the pluripotent state, as in they were now self-renewing and have the ability to differentiate, and we call them stem cells, pluripotent stem cells. So actually, Gail Martin in her paper, in the abstract, you can even look up in this PNAS paper, uses the phrase embryonic stem cells to describe these cells from mouse blastocyst embryos, or what we now abbreviate ESC, and in the lingo in general people try to distinguish, because they're different, mouse and human embryonic stem cells, a little m in front of it or a little h in front of it for human. So this is the 1981, the initiation of the term embryonic stem cells, and you learned yesterday from Gustavo about reprogramming and the engineering in the laboratory of cells, a very similar phenotype or state called induced pluripotent stem cells that we think of. All the biology I'm about to tell you, despite the subtle differences, iPS, ES cells, all the biology I'm about to tell you, you can just think of as applying to both. All these pathways, signaling pathways, methods, directed differentiation techniques, both. From one species, mouse, ES cells, mice, iPS cells, they should behave the same way. For human, do they behave the same way? Mouse, human, ES and iPS, yes. Do they behave identically to mouse? No. Why is that? For whatever reason, the capture of these cells in the stable state in these culture conditions resolves to a basal state in mice that looks closer to the inner cell mass-like state. And these first conditions for preserving these would involve serum and mouse embryonic fibroblast feeders, that the cells grew on top of, and that would preserve them in this, not identical to, but state close to the inner cell mass. For human, which is the cells you're working with all week, for the human, the biology is not different than mouse, but the stable state these cells assume when you culture a human blastocyst embryo is slightly different. So we can go up all the way to 1998 now. Okay, so we're dealing with the biology for however many years that is, 17 years that I'm about to go through of trying to adapt techniques away from just the black box of serum and MEFs, studying a mouse biology of ES cells, learning a lot. And 17 years later, finally, after a long struggle, Jamie Thompson, in a science paper, publishes the technique for culturing human blastocyst embryos and capturing pluripotent cells that can self-renew as well as differentiate in a stable state in the culture dish, and that is the birth of human ES cells. And what we find is, when we profile those cells compared to mouse, is that for whatever reason, these cells, human ES, assume a stable state that's much more similar to a day or so later in development. Interesting. Okay, so the blastocyst embryo, as it implants in the uterine wall, these cells are moving fast and going to start differentiating. And the first differentiation is a segregation of inner cell mass cells away from cells of this inner layer of the blastocyst, right? This is the fluid-filled cavity. These are the lining cells here. And in the inner cell mass here, these cells become slightly more differentiated. They're epiblast cells. And these cells lining the inner cavity are now the primitive endoderm. And so when you culture human blastocyst embryos and derive human ES cells, they are more similar to this epiblast state than to the mouse or the human inner cell mass-like state. And that means the conditions for preserving these cells, sometimes I've used activin. All our conditions, including the ones you've learned, use FGF2 to preserve them. And you see these are not part of the additives of the mouse. So this doesn't mean mice and human are entirely different species or different biology. The biology is all the same, just the stable state in culture is slightly off in time between the two species, hence they're starting from different states. Hence, when I start to talk about differentiation today, it's really slightly different just in the first couple of days. And then the cells will assume the same gastrulation mechanisms with the same growth factors, but the first initial growth factors you add, they can't be entirely identical because they're starting from slightly different developmental states, okay? Any questions at this point before we move on? Okay, so let's go back to the mouse in that 17-year period of intense study and what's happened since, starting with conditions in the culture dish that involves these feeder cells that the friendliest of which we used for years were mouse embryonic fibroblasts, and then these kind of domed-like colonies of mouse ES cells sitting on top of them. And we're talking about the years before 2006 now, so we're not, mouse IPS haven't been discovered yet. We're just talking about mouse ES cells. So the evolution of the biology was trying to understand what controls self-renewal versus differentiation in these cells, and trying to develop more precise conditions that didn't involve serum and might not even involve feeders. So we're going to go over here to a mouse cell, one cell here. And these cells, turns out, are secreting a lot of stuff, but one goodie that is really trying to have the cells exit pluripotency is FGF4. It's being secreted by these cells. It's binding FGF receptors either on the cells nearby or in autocrine manner on their own receptors. And this is trying to initiate a cascade in the cells of phosphorylation of ERK and MAP kinase signaling. And this is a very strong drive for the cells to differentiate. So continually these cells are trying to exit pluripotency, and something here is trapping them in a stable state, but it's a yin and a yang of trying to leave pluripotency, and this pathway by default almost is neuroectoderm. So this is a strong FGF-induced MAP kinase push to go into ectoderm, and specifically neuroectoderm, and this is why probably neuroectoderm was so advanced and was for many decades now has been successfully produced either in primitive states or now more and more differentiated, advanced, mature neuronal states, whereas most of us trying to make lung, we're nowhere, and definitive endoderm is nowhere on the horizon and not possible. And the cells, you can almost forget to feed them and they would start making neuroectoderm. That was pretty easy. So this is the state of the cells, and the first observation is what could replace MEFs and serum to prevent this kind of spontaneous differentiation that was a problem in the field for a long time. So it turns out that LIF could be added to these cultures, leukemic inhibitory factor. And this would stimulate STAT3 signaling, and it could replace MEFs, okay? So that's observation number one. Observation number two is that in the serum, BMP4 could replace the serum, LIF replacing the MEFs, and this then would be the first serum-free, feeder-free culture conditions that were published to maintain mouse embryonic stem cells in the undifferentiated self-renewed state without too much leaving of pluripotency, without too much unrestrained MAP kinase signaling, and LIF again was acting via phosphorylation of STAT3, and BMP is acting through a variety of ID downstream transcription factors, with obviously phosphosmad 158 must be part of the upstream signal here. So there's the biology for a few years of the field advancing to the point where we had defined conditions, understanding mouse embryonic stem cell pluripotency, the paradigm at that time, which is about to change in the next minute from what I tell you, was that pluripotency therefore must be this restrained kind of state that you need active extrinsic signals to control pathways to maintain pluripotency. Along comes a Nature paper by Austin Smith that really shatters this paradigm and challenges our understanding of pluripotency as follows. So Austin Smith says, He discovers conditions which we now call 2I, with two inhibitors. One inhibitor blocks MAP kinase ERK signaling, and another inhibitor blocks GSK3 or GSK3 beta. Okay, and what Austin Smith discovers is that if you inhibit this kind of FGF4 ERK MAP kinase induced push to differentiate into neuroectoderm with the MAP kinase inhibitor, and you increase the overall growth well-being of the cells, and any residual push to differentiate through inhibiting Wnt signaling, or actually it's activating Wnt signaling or inhibiting GSK3, you could now dispense with everything. You don't need LIF, you don't need BMP4. He was criticized for saying that you might still have residual phospho-STAT3 signaling. That's really part of the pluripotency push, so he repeated this work in STAT3 null mouse ES cells to show, in fact, you don't need any active signaling. So his contention that has set the bar for the field is that pluripotency now is this basal state that in the absence or devoid of all extrinsic signals, you can have a cell just self-renew and maintain pluripotency. That was really an interesting Nature paper, and it's a shocking kind of realization that maybe pluripotency is much more self-sustaining than we thought and relies on the absence of all extrinsic signals. I'm going to stop there with pluripotency. Any questions? So when you're playing with the cells in the culture conditions, you're studying human cells that are maybe a little more epiblastic than inner cell mass-like, and FGF2 is in the supplementation. We do also have mouse cells in the center, and we have played with 2I conditions, and it's an area of evolving literature to establish the same constellation of inhibitors. How many are we up to for human? 5I, 4I, 5I. You know, we don't have 2I conditions for the human, but there are some prominent labs, including Austin Smith, Jakob Hanna, Rudolf Janisch, really in hot pursuit of following Austin Smith's biology from mouse into humans, with trying to establish the absence of all input to human cells to culture human ES and IPS cells, and those cells should not be. The goal is to not have them be in the epiblast state, but to resolve so that all biology comes back to basal inner cell mass-like pluripotency in the absence of extrinsic signals. We're going to switch to any extra comments?
Is there a price to pay for
Yeah.
these 2I conditions?
Right, so things are, we often get excited when these conditions come out because they seem so simple and elegant, and the colonies look gorgeous and everything resolves to inner cell mass basal pluripotency. And what we have found in our center, and maybe this is more gossip than established publications at this point, is that the cells seem to get abnormal karyotypes very fast in our mouse 2I conditions. So this is not free just to take the latest conditions that are seem perfect. They are impressive in pluripotency. I mean, they're so impressive they can rescue lines that are not germline competent and make them germline competent to make new mouse lines. They're so powerful. But the karyotypes for us drift very fast within a few passages in 2I.
How do you know whether you have inner cell mass or epiblast in a dish? Do they look different? Do they stain differently?
Well, the human and mouse look very different. I'm not sure that's the marker or metric we'd use. When you push the human cells back with these new inhibitory conditions I'm talking about, and they make a more inner cell mass-like state, they do start to look more mouse-like. They dome up, and so shape might be one measure, but it's really markers, and these days instead of 2, 3, 4 markers, it would be, RNA-seq of the whole genome, and they look more and more similar to this control versus this control. But shape's one of them, yeah. And then you can use, we'll deal with this more on Friday for characterization in your alkaline phosphatase staining, but this kind of constant attempt to leave pluripotency you can pick up very nicely in the dish by just using a simple alkaline phosphatase enzymatic colorimetric assay. And that's not a specific marker of ES cells, but because it's in the intestine and elsewhere. But in this particular model system, cells in the basal state, or even the epiblast-like state, are bright colored for alkaline phosphatase activity. And as soon as they get even a little bit of too much MAP kinase signaling, they lose this color, and you can monitor it in a dish the way the cells are in their health. So we're going to switch now to exiting we've been in pluripotency, we're going to exit pluripotency. So the first attempt to exit pluripotency is just to chaotically let the cells do whatever they're tending to do. If you withdraw LIF, if you don't have the right feeders, you forget to feed the cells, they will tend to go with what they're trying to do, FGF, autocrine, or paracrine signaling inducing ERK-MAP kinase, and exit to neuroectoderm. Other primitive germ layers like mesoderm and mesenchyme, like weeds, if you have a few cells leaving that way, they will tend to overgrow the dish, like any of our culture systems that involve a fibroblast or mesenchyme. So the first few years, that 17-year period of trying to differentiate mouse cells, part of that time involved letting the cells chaotically start to exit pluripotency, but trying to develop culture conditions that might help them achieve this gastrulation kind of effect to more than just neuroectoderm. And those culture conditions were often performed in suspension culture, so taking these adherent cells now and culturing them, let's say, in just a plastic bacterial culture dish that's a non-adherent kind of petri dish. The cells would float in the, let's say, not even in precise media, just in a 10% serum media, and they would be healthy and grow, and they'd start to form these balls. Okay, and these balls were named embryoid bodies, EBs, because if you profiled the cells in the balls, they would have a decent number of cells making primitive ectoderm, neuroectoderm, expressing neuronal markers, mesoderm, and fibroblasts would come up, and even some FOXA2 expressing cells, right? We know there's 3 germ layers, right? The mesoderm, endoderm, ectoderm, and the endoderm we're interested in for this course is the definitive endoderm, and one of the markers is FOXA2, the classic endodermal marker. So is there endoderm in the embryoid bodies? We keep talking about the other 2 germ layers. So there was definitely FOXA2 positive cells profiled in the literature during those years, and it turned out the marker constellation, the potential of those cells was they were extra embryonic or primitive or visceral endoderm, no definitive endoderm whatsoever. Okay, so no definitive endoderm to any detectable level in early chaotic embryoid body cultures. So this is really not going to meet the needs for our course, right? So to introduce the topics for this week, we'll start to talk now about directed differentiation, how to take these kind of old methods that were chaotic and serum-based without controlling defined factors and start to precisely have these cells exit pluripotency towards one germ layer of interest and the downstream endoderm, which is what you'll start to make today. So to do this, if it's directed differentiation to recapitulate in vivo developmental biology, we'll start to draw how the endoderm forms in the embryo and then try to recapitulate that in the culture dish. So in the embryo, if we fast forward up to E6.5 to 7.5 of development during gastrulation, the embryo is going to form with the epiblast, cells of the epiblast starting to migrate down this side of the embryo. And we'll start to draw some axes now because we're going to have asymmetry in the embryo. So we're going to name this pole anterior and this posterior. And this will be caudal, rostral up here, and caudal or the tail down here. So the epiblast cells will migrate down this posterior side of the embryo. So here's your E6.5 embryo. And the primitive, so we'll just label this primitive streak. And then this portion of the primitive streak here we call the anterior primitive streak because if this is anterior pole, anterior posterior. These cells will go on to form a definitive endoderm. And after the cells migrate, as they fold, will form the gut tube made of the definitive endoderm, and the gut tube will get patterned. So the earliest moments of primitive streak formation and then patterning to anterior primitive streak we'll review now and then try to recapitulate those in this cultured cell. We can detect primitive streak because it expresses brachyury, and so brachyury is a pan-primitive streak marker that comes up in all the cells. We can start to detect anterior primitive streak from posterior primitive streak. So if we just divide this in half, this is PPS, posterior primitive streak, and anterior primitive streak. Because anterior primitive streak starts to express FOXA2 together with brachyury, whereas posterior primitive streak has lower to negative FOXA2 and just expresses brachyury. So these two markers we'll keep in mind as we do the following experiments. Gordon Keller published in a development paper a technique to make endoderm utilizing a brachyury GFP knock-in reporter to illustrate the technique. So we'll do this experiment that Gordon originally did together to see how primitive streak is made. Let's take this mouse embryo that's developing that has brachyury, GFP knocked into the brachyury locus. So the embryos are not green, and if you start to make mouse lines and harvest the mice at E7.5, you can see in these embryos a bright green primitive streak from the GFP reporter. If you take, the mice are made with knock-in ES cells, right? So if you take the mouse embryonic stem cells that were used to make this mouse, where the reporter is working, and instead you culture those cells, off of feeders, Gordon screens some candidate factors that he postulates can make endoderm, and he tests them on these cells to see if he can start to induce the green reporter. This is the first endodermal screen I'm describing to you. So from the embryo, the hint is that in this area of the embryo, the developing node is cells secreting high levels of nodal protein. And what nodal protein is doing is it's binding activin-like receptors, other receptors that induce TGF-beta signaling. TGF-beta signaling means the phosphosmads 2 and 3 are being activated. And this is how the embryo forms anterior primitive streak. These cells see the highest concentration of nodal, and it turns out this is what induces those cells to go on to definitive endoderm. So at the time Gordon was doing this paper, nodal protein wasn't easily available to him, but the surrogate that binds almost all the same receptors as nodal, activin A, was. So he used activin A in his screen, and he found that high doses of activin A push these cells to become brachyury GFP positive. Okay, so if you actually go through, because you're going to do FACS this week, if we go through a FACS experiment of these cells, the first experiment looks like this. If on this axis we're, let's say we're just doing an autofluorescent channel here, some red channel, and on this channel we're doing GFP to track the brachyury GFP. On day 0, or the undifferentiated state of your experiment, your cells, each cell being a dot by flow cytometry measured, will be negative or non-green. And within 4 days, day 4, one would observe a lot of these cells turning GFP positive like this. Okay? This is in mouse ES cells. So if we track a kinetic from day 0 to day 6, with brachyury GFP being scored in replicate wells in response to activin, the kinetic will be something like this, okay? And if you do the parallel wells with no activin, the kinetic would be like this. Yeah? So this is a controlled experiment. This would be the actual result, where you don't get induction of this reporter without activin, but in presence of activin, you peak at the frequency, the percentage of cells expressing this marker, maybe 50% or so in the mouse, or 80% in the mouse, and then a decrement. So what's going on, and what does the decrement mean? So if you profile the markers in those cells, they seem similar to primitive streak. And if we introduce our other marker, FOXA2, as I'll draw it for you now, we can learn a lot more about what the cells are doing in response to nodal signaling or activin. And this is really what you're going to be doing during this week. You're going to be initiating primitive streak in the cells starting today and adding activin to try to push your human cells through the same primitive streak or brachyury induction. So are you making endoderm in this experiment is the question next asked. So we have a, a two-reporter system now published by Gordon Keller. So what he chose to do was put into the FOXA2 locus, these are mouse cells, remember, a mutated human CD4 cassette. What does that mean? It means you just have a protein that can be recognized by an antibody. It's a human protein that won't be present in the mouse. And so if any human CD4 traffics to the cell surface, he can pick it up with a colored antibody. You don't have to permeabilize the cells or stain anything inside. That means the cells are still alive. You're just detecting them with the antibody. So let's make the antibody, let's say, red for our purpose of discussion, because we still have the green brachyuria reporter in these cells. So the experiment goes like this now. Day 0, day 6, mouse ES cells that are faxed for FOXA2, human CD4 reporter. Day by day, there's nothing in the undifferentiated state. At day 4, when we reach this peak of activin, actually we do have a signal. And then as the brachyuria comes down, interestingly enough, this stays flat. So what's going on here? What does this mean? So if we actually draw out both axes for you, so we did brachyuria GFP on this axis before. Can I have a quick show of hand who's done fax before, or interpreted fax at least? Raise your hand. Okay, anybody non-interpreted fax? Okay, so fax is basically you're stimulating the cells with a laser to make the molecule, the fluorochrome, fluoresce, and then with the detector tracking the fluorescence. And if I have one cell that fluoresces with both colors, it's going to be up here. If I have a cell fluorescing with neither color, it's down here, with this one color up here, with this one color out here. And so you're getting cellular resolution data over thousands or millions, typically 10,000 cells. And this is what you'll be, I think, expert in by the end of the week. You'll be experiencing it with Dylan and Finn. So if on day 0, none of the cells are expressing these two reporters, they're down here. On day 4, in response to activin, the cells will move up so that a fair portion of them are co-expressing. Some of them are still lagging behind. And then on day 6, I think the interpretation of this is what? These cells are what analogous to the embryo? The interpretation of co-expression of brachyuria and FOXA2.
Should be definitive endoderm.
One step before? Anterior, yeah. So I didn't tell you what definitive endoderm expresses, but anterior primitive streak is the first time you'll see co-expression of brachyuria with high FOXA2. So this turns out is anterior primitive streak-like. So this has been published by a number of groups, including ours, and you can do global transcriptomic profiling and see similarity, not identical, but similarity to real in vivo anterior primitive streak. Day 6 is really informative. Day 6, most of the cells have moved down here. So the cells move this way. There might be a few stragglers here, but this here is a kinetic. This is really important. This is what we would interpret as FOXA2 high, or positive, and brachyuria losing brachyuria low or negative. So these cells drifting here is a kinetic that resembles anterior primitive streak moving to definitive endoderm that has a phenotype that's now brachyuria in vivo, brachyuria negative, FOXA2 positive. So it turns out that in vivo, really only the anterior primitive streak co-expresses these two reporters. So if you recreate this in vitro, it's very likely you have something unique, and that's in fact validated through the transcriptomic and other analyses I told you. And the kinetic, just in response to one single factor, activin or nodal, works the same way, is to have cells efficiently and rapidly, within 4 days, form anterior primitive streak, within 2 further days to form definitive endoderm. And this is the first definitive endoderm published by Gordon Keller, in development in, I forget what year now, 2007 maybe. And it solves the problem, long-standing problem of having primitive or visceral endoderm only in the EB serum-based system. So this really was a landmark discovery that opened up everybody's projects who were interested in definitive endodermal downstream organs of the gut, liver, lungs, pancreas, thyroid. All our work is based on the effect of nodal or activin signaling, allowing the first cells of that germ layer definitive endoderm to be engineered in vitro from mouse embryonic stem cells and later from human embryonic stem cells. Okay, so that's a couple of experiments. Now let's just repeat what we said conceptually with some cartoons of gradients of these growth factors, including nodal, laid over that gastrulating embryo. And this conceptually is really important for how you do your work this week. Go ahead.
What's forcing the loss of brachyury? So you get the expression of FOXA2 positive, and then
Right.
it loses it. Is there another growth factor that's causing the loss?
Yeah, I don't know. I mean, we don't know, but the only variable we're controlling in this experiment is time. So activin and time. So obviously there must be cascades as the cells go to each sequential sulfate decision, but we're not exogenously controlling or inducing that. So I don't know what the variables are.
Okay.
Okay. So let's come back to that gastrulating embryo with the primitive streak here. Again, anterior, posterior axes here. There's some extra embryonic cells on the outside here of the streak. So the streak's formed by migration, again, of the epiblast cells coming down here. Anterior primitive streak, again, is FOXA2 high. Posterior primitive streak is FOXA2 low or negative. But all the primitive streak is brachyury positive. Definitive endoderm is going to form from this portion. The posterior primitive streak migrates to form the mesoderm, and that'll give rise to the hematopoietic system and the musculoskeletal system. So if you want to make mesoderm, you want to also make primitive streak, but there we would not want anything drifting towards FOXA2 high, so nodal signaling might be a problem. So we're very focused on the gradients of how much of each signal you see for how long. So the gradients that work here at this stage in gastrulation around E7.5 can be drawn out as follows. We've just drawn out that nodal comes from the developing node right here. So the highest concentration of nodal that any cell would see in this embryo is down here, and less as we move rostrally. Okay. And that's clear from these experiments, right? That's one factor. So of course there's many other factors at play here. From these cells is secreted Wnt, so there's a Wnt gradient here for canonical Wnt signaling. And there's a gradient being secreted from here, that's BMP4, BMP signaling. So these are the three classic gradients I'd like you to remember, and if you just keep conceptualizing these, I think you can figure out a lot of how to make different cells during directed differentiation. It turns out that primitive streak first forms before you have any patterning or FOXA2. The earliest formation of primitive streak requires TGF-beta signaling. So nodal at low levels will start to induce this signaling and will form streak. Wnt also is a very strong driver of streak formation. And you can activate downstream BMP. So if you add BMP alone, you'll also form streak. So any of these three factors will form primitive streak. But if you have, logically from this constellation, if you wanted to design a protocol to make mesoderm, anybody want to take a guess at what mixture of these, if any, you would make? Do you want to take a guess if you wanted to make a mesoderm you had a mouse embryonic stem cell in culture based on these 3 gradients by an experiment, what would you add to mouse ES cells?
BMP4?
Yeah, BMP4 would be a good guess, right? BMP4 is up here. You're going to stay away from anterior primitive street, and in fact, that works. You can do exactly that. You can take mouse ES cells, add 1 factor BMP4 weight. If you profiled brachyury, you'd actually get very nice brachyury induction. If you added FOXA2 to it, you would not. You'd get very low. You'd be flat. You would not induce FOXA2 in those cells, and actually, if you kept the cells in BMP4, you'd get and use, there's a classic mesodermal marker, KDR, one of the VEGF receptors, you'd have nice induction of KDR. So there you go. If you use this, you can usually figure out a pretty good protocol. All right, so how about if you add Wnt? Wnt, you get primitive streak formation, and you might guess that Wnt would not allow you, it's up here, would not allow you to form anterior primitive streak and endoderm efficiently, and that's true as well. So you wouldn't want Wnt for too high or too long if you're making endoderm. Nodal, it turns out, is in a lot of the protocols to make also mesoderm because it's such a strong streak inducer, right, for TGF-beta signaling, but it's used in very low concentrations relative to the endoderm for short periods. So a typical mesoderm, so you got it right, BMP4 alone can be used, and if you want to make it more efficient and better, people would have added, this is published, 24 hours of a nodal dose. That's about 20% of the dose we'd be using there. Okay, so that's how it works. If you track some other interesting gradients here, there's one here we don't talk about too much, but this is DKK1. This is the inhibitor of Wnt signaling being secreted from this side of the embryo. So look how beautiful it is, right? Wnt signaling inducers here, the inhibitors opposite. And you can see the patterns here of rostrocortical signaling. So noggin and the mutant gives you phenotypes at the head. Right, that's why it's named noggin, the inhibitor of BMP4. So this all kind of works. And Wnt in general, Wnt is, this is posterior here, anterior posterior. So in general, Wnt is a posteriorizing agent over and over in developmental biology. At this stage I'm showing you, but after you pattern and form the gut tube, Wnt would be a posteriorizing agent again. So repeatedly these patterns come on in developmental biology of dorsal ventral, anterior posterior, Wnt being a classic posteriorizing agent. Okay, on to definitive endoderm, which is what you're doing today. So we told you about how this works in the mouse. It works the same in human, it turns out. Now, human cells from the epiblast stage don't necessarily respond exactly the same way to these growth factors. So activin is actually part of self-renewal biology in human epiblast and ES cells. So we tend not to use activin alone, although it would work, for making human endoderm. So what you'll start using today is an endodermal kit that has these factors pre-worked out, and we've done this in experiments where each factor was added individually, and it's just much more convenient for you to use a kit. It's inconvenient because they don't have all the recipes on the kit, but what you're in effect going to be doing is early, as your cells exit pluripotency, you don't want them going from FGF ERK signaling into neuroectoderm. You don't want that. So in the kit's going to be some Wnt and BMP, some nodal to get the streak going. BMP4 is a potent suppressor of neuroectoderm at this stage, so that helps suppress the lineages you don't want. So Wnt and BMP early, and then you're going to be dealing, in your kit, you're just going to be going and seeing the sustained effects of high activin A nodal signaling. Your human cells do not have brachyury, GFP, and FOXA2 CD4 in them. So how are you going to know if you make endoderm? You can sacrifice the cells and measure FOXA2 by qPCR, but that won't tell you how many of the cells are making endoderm. You could stain the cells for nuclear protein for FOXA2 and count them, and that would be a good measure of how much endoderm you might be making, but then the cells are dead, they're fixed, and that doesn't seem very convenient, right? So we need markers of live cells, and flow cytometry is, we just went through some examples of how powerful it is at revealing what your cells are doing, and the cells are still alive and can be put back in culture. So your cells need to be profiled in some way that doesn't require every time a brachyury, GFP, FOXA2, human CD4, or whatever knock-in reporter system. So early on in the field, people making endoderm would take the cells and would profile them by arrays. And they would find on the arrays are differentially expressed genes that are isolated to the endodermal compartment, maybe not in undifferentiated cells, not in the cells you don't want. And you could restrict that list of hits to just cell surface proteins. And 2 of those hits, and then you can start buying antibodies for those hits and see which ones actually identify your cells by flow cytometry. And 2 hits that were validated in the literature, c-KIT and CXCR4, help to monitor the efficiency of differentiation of your cells toward endoderm. Okay, so let's just pause here for a minute. Look at these names, c-KIT, CXCR4. You've all heard of these, right? c-KIT is the receptor for stem cell factor. CXCR4 is the receptor for SDF1. What does that have to do with endoderm? Why are we profiling these? Are we adding stem cell factor and SDF1 to our cultures? No. So what are these doing? These are not, this is not very biological. It's simply array hits that worked out that antibodies can score how many cells are positive for these. If you look at the developing embryo, these markers are all over the place, right? In the blood, in the stroma. They're not endodermal markers in vivo, but in this model system, cells in the undifferentiated state are not expressing CXCR4, and cells after activin have expression of these 2 markers up here in the endoderm, not in the other compartments. So they're very convenient only in this highly engineered artificial in vitro model system. Okay, so when you go out on the road and back to your own labs, please don't think of c-KIT and CXCR4 as definitive endodermal markers. They are those markers just in this in vitro system. Okay, you can't slice an embryo, stain for CXCR4, and find the endoderm. It'll be in the endoderm and everywhere else too. Okay, is that clear? Okay. So to profile, we have an early paper in mouse by Constina Christodoulou in the JCI that provides in mouse an overlay day by day over 6 days of the activin response of these markers compared to these markers, and it is just it's a useful resource if you want to see laid out visually how all the markers move over time in response only to activin and how valuable c-KIT and CXCR4 are. So what we're going to do with you guys, this is starting today, right, Marianne? Yes, okay. Is we're going to take your cells, your beautiful cells that were growing the undifferentiated state, somewhere between epiblast and inner cell mass, those colonies you're looking at. Which cells are we using today? What line?
For the definitive.
For the, yeah.
I don't know what Dylan's using.
Which line is, okay. So he has some human pluripotent stem cell colony that you're now going to enact the protocol of having them exit pluripotency, but in serum-free conditions, only adding these growth factors through this endodermal kit. You're going to push them to recapitulate this kinetic. The kinetic will be faster in these human cells that I'm showing you in the mouse here, so that rapidly you're going to, if you faxed, let's say for CXCR4 c-KIT on day 0 before they differentiate, what you'll find is they actually are c-KIT positive, they're dim, so they'll be around here, and CXCR4 negative. And then by day 4, in response to high activin signaling, as they go through the same kinetic, which you won't be measuring, but they go through the same kinetic, you'll be able to see the efficiency of that differentiation by faxing using fluorescence conjugated antibodies to these 2 cell surface markers. The c-KIT will rapidly have dropped and then come back even brighter, so it'll be out here, but also the cells will have turned CXCR4 positive. So these cells will be here, measured by flow cytometry. There'll be a few stragglers that are either dimmer or negative, and the percent of cells in here, that's typically, I don't know, 80+%, sometimes even close to 100%, will give you, monitor the efficiency of your directed differentiation, your response to this sequence of gastrulation, starting with primitive streak to anterior primitive streak to definitive endoderm. So you want to make lung next, right? Okay.
So is it possible to maintain them in this definitive endoderm and expand them like that?
Yes and no. Yes, there's a paper by Paul Gadou about endodermal progenitors being maintained in a stable state once they've reached the state.
Mm-hmm.
You can pick them up
So
as colonies and you can expand them and freeze them and thaw them.
Yeah.
However, his goal was to make liver and pancreas, and so the cells I don't think have the as broad a potency or differentiation repertoire as these exact cells. So what he's able to do is capture, you know, a liver, pancreas competent, probably gut competent also, subset of the definitive endoderm. Okay, we haven't really established yet a capture of a lung competent endodermal progenitor that's self-renewing and stable. Okay, so let's make these lung competent. We can go to the developmental biology literature now that we have definitive endoderm and look up papers that might suggest the key inducing factors for lung cell fate. So you can come up with names like, I'm going to erase this now. Okay, we got this. I'm going to erase this. And we need some markers. So, start with the marker first. If we have the endodermal gut tube and we just unfold it. Okay, so this is around, let's say, E 8.5 to 9. If we unfold the gut tube, this is supposed to be a tube. Not a great artist, but there. Okay, that makes it a tube. You can find FOXA2 everywhere. That's our pan-endodermal marker. And then we can find some, like we did with the anterior perimeter streak, we can start to find some other markers that are not pan-endodermal, that are more regionalized, that tell us patterning. And finally, we can find some not entirely specific, but maybe enriched or highly specific transcription factors that identify cell fates. So under the microscope, everything looks the same, but with transcription factor staining, you can start to find organ domains like hex for the liver, NKX 2.1 for the thyroid and lung, PDX1 for the pancreas. The patterning markers are as follows. SOX2 is the classic anterior foregut or anterior gut tube marker. CDX2 is the classic hindgut or posterior gut tube marker. Okay, so it's almost like binomial. Around the pancreas, these two markers meet. So this side is CDX2 positive, SOX2 negative. This side is SOX2 positive, CDX negative. Within the SOX2 foregut domain, then, you've got these domains that are about to bud and differentiate, or in the case of the lungs, bud, branch, and differentiate, NKX 2.1. Okay, so if we go back to our definitive endoderm about to make lung, and we look at classic markers, SOX2 will not be very high. And there might be quite a bit of CDX2. If we look for our favorite marker, NKX 2.1, are any of the cells, the endodermal cells, positive for NKX 2.1? No. So NKX 2.1, as well as these two markers, are pretty ideal. And
Sorry.
that's okay. It's probably Nico calling his mom. Right. Okay, so NKX 2.1 is a pretty perfect marker because it's off here, and all we have to do is add some growth factors to induce lung, turn on NKX 2.1, provided it's endoderm and not neuronal NKX 2.1, and we're pretty likely to have either thyroid or lung, right, based on these domain markers. So if we look up growth factors that induce lung, you know, Wnt, canonical Wnt inducer is all over the literature, and BMP, like BMP4 is the ligand, BMP signaling is all over the literature, maybe retinoic acid, and there's some debate about FGFs. But you can quickly find candidate growth factors to add to these cells. All you have to do is profile the induction of NKX 2.1, and you get nothing, no lung. That makes no sense, right? There's no lung. So we did this for years and years. Nothing makes lung here. So something's very wrong in the system. And Hans Snook is the investigator who really made the discovery that solved this problem or hurdle I just described to you. So he had a Nature Biotechnology paper that came as follows. He was trying to make thymic epithelium because he was doing a hematopoietic immunological differentiation project, trying to make T cells, needed thymic epithelium to put with those. He knew that thymic epithelium came from the foregut endoderm, tried to make endoderm, human ES-derived endoderm, into foregut to make thymus, and by mistake he made lung-competent foregut endoderm. Okay, so his serendipitous discovery blew our field open, and we welcomed him with open arms to the lung field, and that Nature Biotechnology paper is our key paper we refer to that solved the problem of these cells not making lung. And the problem was as follows. The cells were not, they were expressing too much of this, not enough of this. They were not foregut, they were just definitive endoderm. And so Hans's discovery, in a nutshell, was to anteriorize the definitive endoderm to make anterior foregut endoderm. And the way he did it was to inhibit BMP inhibition, and inhibit TGF beta signaling. The effect was SOX2 went up, CDX went down, the cells were foregut, and now if you start to add the same exact factors that didn't work before, suddenly they work. The endoderm is rendered competent now in the presence of Wnt, BMP, and RA. So the Wnt is actually stimulated through a molecule called KIR, which is a GSK3 inhibitor. GSK3 beta inhibition stimulates canonical Wnt signaling, activated catenin goes to the nucleus, and in the company of BMP4 and retinoic acid, out comes lung, and that's measured because NKX 2.1 comes up, that nice lung endodermal marker. The concept here to understand is that every cell fate decision in a cell depends on what it's just done immediately before. Okay, so if you don't anteriorize this, you'll not get lung, even if you're using the right factors. If you don't make anterior primitive streak, if you make posterior primitive streak, too little nodal, too much Wnt, it's going to want to be mesoderm. Now you put it into these inhibitors to make anterior foregut, it'll do something totally different, will not respond and make lung. So everything depends on the step before it working properly. That's why it's critical today that you learn how to measure, monitor the efficiency of the early endoderm, and you check it every time, because the cells will tell you on day 4 if they're inefficiently responding, and then you don't want to go to day 30 and 40 making lung when the cells would have told you they're making poor endoderm. They're not going to behave. But because you were in a rush and didn't check, you didn't know, and because you didn't want to spend the extra hour, you wasted 30 days. So I just encourage you, when you can, ask your cells, ask them how they're doing, they'll tell you, and feel free to abandon experiments when things don't go right. You know, the question is always, did the staining not work and the cells are fine versus are the cells not fine? We debate that all the time. So today you go through the protocols of how to stain, how to monitor, and then hopefully you can build confidence to do the technique yourself and trust that the cells are telling you they're on track or not on track. Okay, so to recap again, if you come away from the week with techniques of how to, you have some understanding of reprogramming, techniques to maintain your cells in the undifferentiated state, understanding of the biology of how they're being maintained, and then to control them for directed differentiation and leaving pluripotency in a very directed way, not that chaotic early serum-based way, but in a way that makes primitive streak, mainly in response to either Wnt, BMP, and nodal, but then anterior primitive streak in response to nodal signaling stimulated by activin A, followed by, with the passage of time, definitive endoderm in response to activin A, followed by patterning of the endoderm to anterior foregut endoderm with BMP and TGF beta inhibition as discovered by and published by Hans Snook, followed by actual induction of lung fate. It would be nice to visualize the lung fate with seeing NKX 2.1 come on, and that's what we have a knock-in line, NKX 2.1 GFP knock-in in the human system, where you can see the green, and Dylan and Finn are supposed to show you that by the end of the week, so you can see how you monitor lung fate in response to those conditions. Questions? We're out of time, so I should stop and just let you ask a few questions, and then Marianne will take over.
So once you get to that NKX 2.1,
Yeah, right.
how far do you go here in the facility or in the?
Well, the goal is to make all the lung lineages with increasing maturation.
Mm-hmm.
And we had a stem cell paper 3 weeks ago or so that showed how you take these through the lung progenitor stage to airway maturation, so proximal airway epithelium, including multiciliated cells and secretory cells. And then we have another project trying to make them distally, which goes pretty well, and then so SOX9 is a marker of distal fate, and then to mature them all the way to a functional type 2 pneumocyte is our latest project, and that's also advancing pretty quickly, where some of the cells in distalizing conditions make lamellar bodies and process surfactant. So I don't know if that answers your question. I think that basically we're part of the way there, a bit better each year, and a long way to go in terms of very postnatal, functionally mature adult cells is the ways to go. Yeah.
mean, the literature is just so fluid now. People are using ALIs and 3Ds and co-cultures with this and that. And
Yeah, I think the goal is when you have really postnatal advanced primary cells, they should behave in all the standard primary medias from ALI to 3D. And if you don't make the right cells, they're always going to look funny, right?
How sensitive are the cells for the day? So you have at day 4, you express these markers. If you leave them for day 5 or day 6, do they change?
They change. Well, the markers, yeah, change less rapidly than the competence of the cells. So yeah, we don't mess around too. So we always check, and if it's not working, it doesn't mean the line's bad or something's failed. They might need to be rechecked 12 hours later, and they're coming up in endoderm. So there's line-to-line variability based on genetic background, less clone-to-clone variability, but even in the same clone that's behaving, for reasons nobody really in the country understands. I don't know if it's media serum, not serum, but media batches, growth factor batches, the humming, buzzing in the building, it seems to have run-to-run variability. It's a problem. That's why we really like to check the efficiency. Yeah, changes fast. So when you're learning this and practicing, if you want to go home and practice, I think the way we recommend, because every line is different and it does fluctuate quite a bit, get a line you know makes lung that works, particularly one with a reporter is the best way to always get day-by-day visual feedback of whether they're turning green or not, if NKX 2.1 is your goal. And then you can learn a lot. If you take you make your own line new and start to work with it without any control that you know works, you can really waste many months with a bad line or inefficient line or